This video is on colon cancer. Colon cancer. And colon cancer is relatively common. In fact, it's the third most common cause of cancer death. Third most common cause of cancer death. And because it's so common, causes so much death, we want to screen for it. Yeah, we want to pick it up early. So we can screen for it. We usually start at around 50. So once you're over 50, you'll do colonoscopy. Or if you don't want to do those invasive procedures, then you can do things like stool samples. You can send stool samples, and we know that the tumor can bleed or shed some of its mutated cells, and we can look up the stool for blood, things like that, DNA. So I'll just write stool, blood, DNA, and those are just some ways we can screen for colon cancer. All right, try and pick it up early. And if we do pick it up, then we'll just resect it, chemo, regular cancer stuff, regular cancer treatment. And we want to monitor the treatment, make sure it's going well. We monitor it with a blood test called CEA. CEA stands for carcinoembryonic antigen, and it's made in your GIT, and it's not specific to colon cancer, so we can't use that test to screen people. However, if we have a baseline measurement and we give them treatment, then we hope that CEA goes down. If CEA doesn't go down, then we know something's going wrong. So CEA is good for monitoring it. Monitor. So just know CEA is a great way to monitor colon cancer, commonly asked. Now CEA, as good as it is to monitor colon cancer, CEA can also deposit into your kidneys. Deposit in your kidneys cause nephropathy. Do you remember what nephropathy is associated with cancer? It's a very common one. That'd be membranous nephropathy. Remind me, is membranous nephritic or nephrotic? Give you a second to think about that. Nephritic or nephrotic? It is nephrotic, so that's the proteinuria. Okay, good. And so that one was associated with cancer, and now you know why, because CEA can cause membranous nephropathy. So this is all screening, this is all treatment, but for those that don't screen, then they might develop and not ever know it. And that tumor can grow and grow and grow. And when the tumor grows, it, one, likes to occlude the lumen because it's growing in size, two, likes to bleed. Okay? So some signs that they might show up to your clinic with is obviously things like weight loss, fever, you know, all those constitutional symptoms, but also obstruction and bleeding. Obstruction and bleeding. And it's most common in the recto-sigmoid, so really at the end part of your whole GIT tube. So your recto-sigmoid, but if it doesn't affect that, then other places are your ascending colon and your descending colon. Ascending, descending. And like I said, the signs are obstruction and bleeding. Sometimes they like to say the left side bleeds, right side obstructs. Why do they say that? Both of them bleed, both of them obstruct. Why do they say the left side bleeds, the right side obstruct? Because your left side is a smaller caliber. Yeah, that's where your poo exits. So your left side is a smaller caliber. So the first sign you'll notice, because it's a smaller caliber, is obstruction. Once that mass grows, it's the first thing you notice. Obstruction. If you do a barium swallow, you'll see that the gut is very, very small. They sometimes call it a string sign because it's so obstructed. So string sign. Sometimes you get pencil thin stools. Yeah, the patient will present or complain of pencil thin stools, and that's because the stool gets pushed through that obstructed lumen, comes out pencil thin. So, all right, pencil thin. We said the right side bleeds. Why do we say the right side bleeds? Because your right side is a larger caliber lumen. All right, so that tumor can grow and not obstruct, not cause any kind of symptoms. You won't notice anything. The first thing you do notice, however, is that it'll keep bleeding, and then you'll get iron deficiency anemia. You'll get bleeding in your stool, you'll get tiredness, you'll get exertional dyspnea from the anemia. So the first sign you notice is not going to be obstruction because you have a large caliber right side. It's going to be from that bleeding. That's why we say the right side bleeds. So left side obstructs, right side bleed. That's why I said, but now you know. Another thing they can present with, other than obstruction and bleeding, they can present with bacteremia. Whether it's bacteria growing on the mass, the tumor, or disrupting your gut and causing increased bacteria. Bacteremia seems to be a common thing. We talked about this way back when, cardio. We talked about infective endocarditis. And you can have a bacteria that goes into your heart, causes infective endocarditis that's very associated with colon cancer. Do you remember that bacteria? Hopefully you do. That'd be group D strep, aka strep gallolyticus. Strep gallolyticus, formerly known as strep bovis. They don't call it that anymore. Strep gallolyticus. Just a micro throwback. Commonly asked to talk about someone with infective endocarditis with strep gallolyticus. What's the next step you do? Colonoscopy, because they probably have colon cancer. Now, what are the risks of developing colon cancer? Well, risks include things like IBD, inflammatory bowel disease, because you just wreck your bowels, have a lot of high cell turnover, inflammation, stuff like that. Smoking, because why not? Smoking causes every cancer, it seems like. Processed meat. There seems to be carcinogens in processed meat. You can have hereditary cancer syndromes, where you inherit some mutated gene and you just have an increased risk of cancer. You can have hereditary cancer syndromes for any of your body parts. So hereditary cancer syndromes, and if you have polyps, what the heck are polyps? Polyps is a collection of tissue that you might see in your colon. And it's those polyps that can become cancer. You don't usually go from normal to cancer right away. You usually have like hyperplasia, you get some mutation, you get some dysplasia, and then you get cancer. So polyps will show up as this increased growth of tissue that can undergo dysplasia, become cancer. So if you have a lot of polyps, that's a risk. And in fact, that's what we screen for when we're doing colonoscopy. We look for things like polyps, and things like cancer obviously, but things like polyps. Now, how does your normal epithelium become so mutated? How does it make these polyps in your colon? How does it become so mutated? There are many different pathways. Let's talk about some of them here. The first pathway is if you silence or mutate your tumor suppressors. That's a no-brainer. You silence or mutate them, then you silence or mutate your tumor suppressors, then you won't be able to suppress tumors. Tumor suppressor genes that you should know is APC, so mutations of APC can cause it. You can have something really, really cool sounding called hypermethylation of CpG. Hypermethylation of CpG. What does that mean? In your DNA, you have these islands of cytosine and guanine, your nucleotides, and these islands are usually clustered around your promoter. Yeah, that's what helps gene transcription. Methylation silences things in biochemistry. So if you silence those islands and you silence promotion, you silence DNA transcription. And that way you can silence your tumor suppressor. Just a fancy way of saying it, but hypermethylation of CpG sounds pretty cool. Another mechanism you can have of turning your normal cells into mutated cells will be turning on oncogenes. Again, that makes sense. An example would be BRAF. BRAF is an oncogene, and if you have a mutated one, then you can have cancer. Usually we associate BRAF mutations with the two-hit hypothesis. What's the two-hit hypothesis again? You have two alleles, one from mom, one from dad. If one of them goes wrong, one of them gets a hit and goes out, that's all right. We still have one allele to kind of cover for us. If that allele goes out, gets hit, two hits, then you have no alleles. Yeah, and then so you'll express the cancer, or you'll express the mutation. All right, so first hit, second hit. That's a two-hit hypothesis. If you inherit one that's already defunct, then it'll just become the one-hit hypothesis, yeah, because you're already halfway there. But if you inherit 2 normal ones, then we call it a 2-hit hypothesis. And you have a BRAF mutation. If you have an increase in your oncogenes, then you have what we call familial adenomatous polyposis. You have so many polyps, it's unbelievable. Your colon's like more polyps than it is colon. You have literally sheets and sheets of polyps. So I just write, I don't know what I'm gonna write, polyps. Now, is there a chance that one of those bazillion polyps can turn into cancer? Absolutely. In fact, you're guaranteed to have cancer. So these patients, basically 100% of them have colorectal cancer because you just have so many polyps, and they're so mutated that, of course, they can become cancer. Now, a mutated gene like BRAF isn't just going to cause cancer in your gut. It's not just confined to your gut. That wouldn't make sense. It can cause cancers in other places. And so you can have FAP plus other cancers. For example, you can have FAP plus fibromas and osteomas. So MSK cancers. We call this Gardner syndrome. Gardner syndrome. And some other signs of Gardner syndrome is supernumerary teeth. You have a lot of teeth that can become impacted because there's so many of them. And you can have hyperpigmented retina. Okay, so some questions I've seen on this is that a patient will come in with weight loss, diarrhea, they have supernumerary teeth, they have impacted teeth, they have tooth pain, they have a giant knob basically on their side of the jaw, that's the osteoma part. And then they might ask, what else do you expect to find? If you look into the eye, you might see the hyperpigmented retina. So they kind of jumble all these variables together and make into a step question. All right, so that's Gardner syndrome. You can also have FAP plus brain tumors. Brain tumors. That's called Turcot syndrome. Turcot sounds like turban, which you wear on your head, so that's the easy way to remember brain tumors. All right, hope that makes sense. That's another mechanism where you can mutate your normal cells. A third mechanism is loss of DNA repair. Loss of DNA repair. Again, makes sense why this can cause mutations in cancer. A fancy way we can call this is microsatellite instability. Ooh, that's a fancy term. What does microsatellite instability mean? Microsatellites are these repeating codes of DNA. And they don't change. Every time your cell replicates, they stay the same, so you have these repeating codes of DNA. And that's good. That way we know that your cells are replicating and duplicating as they should. However, if one of the cells replicates and makes a daughter cell that has half of these, then you know something's gone wrong. Yeah. You know you have a loss of DNA repair. Something went wrong in DNA replication, and because there's no repair, it stayed wrong. So if something's wrong with your microsatellite instability, then you know there was a loss of DNA repair. Loss of DNA repair causes something we call hereditary non-polyposis colorectal cancer. And the name gives it away. This cancer doesn't come from polyps. They just arrive de novo. De novo. All right, so one of the few times it doesn't come from polyps. You have a mutation of your DNA gene, and then it'll go from normal to abnormal, and you'll have cancer. Now, is it confined to your GIT? Only cause GIT cancer? No, that wouldn't make sense. So you also have increased risk of skin cancer, and that's not how you spell skin. Skin cancer and gynecological cancers like endometrial and ovarian. So some questions I've seen on it, they'll talk about like a 20-year-old, someone really young coming in with colorectal cancer, and then they say the patient shows loss of DNA repair or microsatellite instability. What is the patient at increased risk for? Skin, endometrial, ovarian cancer, stuff like that. Or they might just say 20-year-old comes in with a history of ovarian cancer, endometrial cancer, colorectal cancer. What's the underlying mechanism? Loss of DNA repair, microsatellite instability. Many ways they can say it, but it's all the same. Our last mechanism of mutation is probably the most common, and that is just accumulation of mutations. You have a normal cell that mutates once. All right, that's not too bad. Starts to accumulate another mutation. Okay, getting worse. Another mutation. Okay, getting worse until finally it becomes cancer. Accumulation of mutations. We sometimes call this the chromosomal instability pathway because your chromosomes are getting mutated. But my favorite name is your adenoma to carcinoma sequence. Sequence means a particular order of things. In this case, a particular order of mutations. Bum, 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 bum. And those mutations will change the cell, cause an adenoma, which is benign. But as it keeps mutating, as it keeps accumulating mutations, it will eventually turn that into carcinoma. So adenoma to carcinoma sequence due to those mutations. Now, what's the order of mutation? I said sequence means a particular order of mutations. What's the particular order of mutations? That is A, K, P, D. Formerly called AK53, but now I like to call it AKPD. AKPD is the order of mutations. The first one to go is going to be APC. Second one to go is going to be your CRAS gene. The last two to go, leave together. That's going to be your P53 and your DCC. DCC stands for deleted in colon cancer, which is a pretty dang giveaway of a name, right? Deleted in colon cancer. That gene, if it goes out, you get colon cancer. Let's talk about this pathway in a little bit more detail because this is one of the most tested and the most common pathways of how our regular cells can go make a polyp, and that polyp can become cancer. You have your normal epithelium, nice and flat, and then the first mutation hits. APC hits. Now the cell's getting a little bit mutated, making this little adenoma. Now adenomas are still benign, all right, we're still okay. However, another mutation hit, CRAS. And that adenoma will grow in size. So CRAS grows it, APC starts it. And then finally, our last two mutations, P53, DCC. P53, something important, something they kind of found out of nowhere. P53, it really seems to rev up COX expression. And if you give NSAIDs, it can actually slow this progression, right? But we'll just say you don't slow the progression. You have P53 and DCC mutation. Those two mutations will turn this growing adenoma, this growing benign adenoma, into full-blown cancer. And that is your adenoma to carcinoma sequence. AKPD. So again, you have normal epithelium, and then APC hits, starts growing the adenoma. CRAS hits, the adenoma starts to grow, and then finally P53 and DCC turn that growing adenoma into full-blown carcinoma. That is your adenoma to carcinoma sequence. Good? So a lot of things can come from polyps, and not all polyps are the same. Not all polyps have the risk of running cancer. All right, some polyps look different. Some polyps are non-neoplastic. Let's just talk about polyps, okay? Let's just make sure we know everything about our good friend polyps. All right, polyps, they can be neoplastic or not. Let's talk about the not neoplastic. Yeah, let's start off on a good note. Not neoplastic, you can just have a hyperplastic polyp. You just have tissue that's growing a little bit too much and is making a little polyp. Hyperplasia is reversible. Yeah, so it's not neoplastic. You can have something like a hamartoma. What's the definition of a hamartoma? Hamartomas are increased growth of normal tissue. It's just a little bit too much of it. So this is just normal tissue. the chances of that normal tissue becoming cancer is not really high. It's just normal tissue, just a little too much of it. However, hamartomas are associated with 2 autosomal dominant disorders. One of them is Peutz-Jeghers. One of them is juvenile polyposis. Judging by the name, you get polyps as a kid. Both of these will make polyps. That's the name of the game. What's the easiest way to tell them apart? Peutz-Jeghers has, remember, that's a hyperpigmented mucosa. Yeah, so they have dark spots on their lips. Peutz-Jeghers, easy to tell apart. Peutz-Jeghers, you have those hamartomas, like I said, that don't really have a chance of becoming cancer. However, that doesn't mean you don't get cancers in other places. So Peutz-Jeghers makes those polyps, but also can increase your chance of cancer in other places. All right, so it's not completely benign. Those are your non-neoplastic polyps. Now let's talk about your neoplastic ones. You can get adenomatous polyps. And they call it adenomatous because they're gland-like. And histologically, some glands look tubular. And this is probably the most common. So you have these tubular glands. So this is your polyp. Your glands would look like a tube. So tubular gland. And these are often pedunculated. So they have a stalk, pedunculated. So they have a stalk, they have tubular glands, but one thing you should know is that they're low risk. Nice, low risk. Sometimes you can have one that makes very villous glands and has a lot of villi. What do you think we're gonna call it? So we're gonna call it villous. Villous. Because it has a lot of villi, it's not gonna really have a stalk. That wouldn't make sense. It wouldn't be like a stalk with a bunch of villi. So it doesn't really have a stalk. Sometimes we call this sessile, which means attached to one place. So it doesn't have a stalk. And these are higher risk. Higher risk. The easiest way I remember is villous sounds like villainous. Yeah, so those are evil. This is evil, higher risk. You can have it in between, we call it tubulovillous, so it kind of has like a mixture of both, and that's intermediate risk. That makes sense. So those are all your gland-like ones. You can have something called a serrated polyp. And they call it serrated because inside looks serrated. The crypts look serrated. Sometimes they call it sawtooth. And there's no doubt that these are high risk. All right, there's just no doubt. In fact, when we talked about our 4 pathways of how normal tissue can become cancer, the first 3 of those pathways, this polyp follows them. All right, so there's no doubt it has increased risk of cancer. So, all right, high risk. So the polyps that you need to know that are high risk, always ask, are going to be your villous, aka sessile, and your serrated polyps. Always ask. They'll talk about a polyp that's seen on colonoscopy and is taken out, and then they might say it's tubular. And they might say, is that high risk? No, it's low risk. Or they might say it's villous. Is that high risk? Yes, that's high risk. Or they might say it's serrated. Is that high risk? Yes, it's high risk. Okay? Or they might show you the histology. If you see the nice tubes, then it's tubular, low risk. If they show you a lot of villi all over the place, then it's villous, high risk. Gotta know that. I've seen a lot of questions on that. And that actually does it for your colon cancers. Now, I just want to close up with a talk on small intestine cancers. There are very, very few small intestine cancers. It's very rare to have cancers in your small intestines. However, you can have something called a carcinoid tumor. Carcinoid tumor. Carcinoid tumors are tumors of your neuroendocrine cells. If that sounds very non-specific, it is. You have neuroendocrine cells all over the place. However, carcinoid tumors commonly show up in the small intestine. And that's rare to have a tumor in your small intestine. So if you see a tumor in your small intestines, think carcinoid tumor. And we know this little mass in this small intestine is a carcinoid tumor because it has chromogranin. Chromogranin. Chromogranin is a protein that's found in neuroendocrine cells. So we see chromogranin, we're like, oh, it's a neuroendocrine cell. We have a carcinoid tumor. And carcinoid tumors love to secrete things. They love to secrete things like serotonin. It's never too good to have too much neurotransmitters in your bloodstream, abnormal excess amount of neurotransmitters. But serotonin, once it's found in the gut, will get taken up in the blood, go to your liver, and our liver is able to break it down. Nice. So we don't see any harm, no foul. What we do see, we'll see the metabolites in our urine, serotonin metabolites in our urine. So we know, oh, there seems to be increased serotonin here. So, but in the classic sense, in the presentation sense, there's no harm, no foul, no real symptoms. However, if this carcinoid tumor metastasizes to your liver, destroys your liver, then you're no longer able to break down serotonin. You have increased serotonin going willy-nilly. And that can cause symptoms. We call all that serotonin syndrome. Serotonin syndrome. Everybody knows serotonin syndrome as things like flushing, diarrhea. What you also need to know, which is commonly tested because they pick a lot of students off because they don't know this, is it causes right heart fibrosis, especially fibrosis of your valves. What are the valves of your right heart? That'd be your tricuspid, pulmonary. Why does it cause right heart fibrosis? Let's draw our little heart diagram. So this is my trusty heart diagram. You have your right heart, left heart. Your right heart goes to your lungs where it gets oxygenated, and that goes to your left heart. Then finally that blood is pumped to your body, and then the venous returns back to your right heart. Okay, so if you have serotonin syndrome in your body, it's going to return to your right heart. And for whatever reason, I haven't found any resource that really explains it, serotonin will go to your right heart, destroy your valves, your tricuspid valve, your pulmonary valve. So that will go and destroy your right heart and go to your lungs. However, in your lungs, you have something that can metabolize serotonin. And so that serotonin goes away, and all you're left with is nice clean blood into your left heart. That left heart goes into your mitral valve, your aortic valve, out to your body. No problems there. So your left heart is spared, but you get right heart fibrosis. Gotta know that for serotonin syndrome. If you know that, I think you're good. That does it for colorectal cancer and small intestine cancers. Hope you enjoyed. Thanks.